understand the concept of molecular diagnosis, the testing, the application, the techniques, and how you can actually bring it home and apply it to the area of your interest based on your expertise. You should also be able to know how to, apply this technique, and then know the quality management that is involved in your samples, how to get them, the kind of quality of the materials to work with. So through the practicals, which some of you that are in Lagos are going to come to the lab, should be able to make you also to acquire the practical skill, to be able to now know how to work on the bench, to be able to do DNA extraction, PCR techniques, basic DNA electrophoresis, to have an idea of the basic concept of molecular diagnosis. Then they can apply it in a larger scale based on where you are working at. So I'll be taking you on module 2, which is the molecular techniques. I am going to give you an outline of some of the techniques you can get in molecular. I think about 12 of them that I have, but I will take time to talk about PCR and then DNA extraction, which we'll do in practical. And we'll also look at the techniques of electrophoresis and how you can read your results and draw inference from each result you're going to obtain based on the conventional PCR, and then how you can now extrapolate and also use that to also apply when you are using advanced techniques and the mid technique that has to do with things like real-time PCR. So my name is Dr. Francisca Nwokori, as I've been introduced. I'm a lecturer at the University of Lagos. So please confirm that you can hear me before I continue. I know I have about 5 participants. Okay.
I hear you.
Okay, that's fine. Thank you. You can mute yourself now. All right. So the first thing I usually discuss with people is for them to understand what molecular diagnostics means as a concept. And I usually try to use this slide or this definition provided by the WHO on what molecular diagnostics encompasses in general terms. So it says that molecular diagnostics is a broad term, of course, is a larger thing, that describe a class of diagnostic tests that assesses a person's health. Of course, you know our job on the bench is to be able to assess person's health using the clinical specimen. But this time around, we're looking at testing at molecular level, and we're able to look at the concept of how to dictate and measure specific genes. So the definition now is trying to let us understand that we are now trying to do testing at molecular level. Molecular level, I want to take this keyword, molecular level, dictating and measuring specific genetic sequences. So what we're looking at when you're doing molecular is to be able to look at gene sequences. So gene sequences can be in single genetic gene you're looking at or multiple genes at the same time. So that gene can be being expressed out of the way you look, or the blood type that you have, or the kind of microorganism that lives in the gut, the vagina, the mouth, or things like that. And if you're interested in clinical chemistry, we're looking at markers that are able to make somebody to have diabetes, or high blood pressure, or some other disorders that have to do with the enzymes. So when we say we're looking at definition at molecular level, looking at specific genetic sequences in what? So this is what I call the molecular tools. So the tools of molecular diagnosis, irrespective of the field where you are, you make use of these tools. So you are using the DNA, RNA, or the protein being expressed to be able to now know the health of an individual at a molecular level by analyzing these 3 tools. So molecular diagnosis, you're either extracting a DNA to work with, an RNA in work with, or to be able to study proteins. So no matter which section you are, whether in clinical chemistry, hematology, histology, you're either extracting a DNA to work with, a RNA to work with, or a protein. So no matter the sample you're working with, whether blood samples, urine samples, hair samples, and whatever. Everything now boils down to this place on these three concepts, which I call the tools of molecular diagnosis. I'm sure you will pick it from here to understand that everything you do in molecular takes bearing from this point. That is your specimen. So irrespective of what sample you're working on, you're going to extract a DNA from it to work with an RNA or a protein. So and when you're doing all that with this association with specific health condition or disease. So if a genetic sequence is in order, normally what is supposed to be, you can say that is okay, is specific health normally. But when a genetic sequence is abnormal, that means the gene you're seeing is not what it's supposed to be under normal body temperature or normal body condition. It becomes an abnormal gene. Then you can use that to say whether the person is healthy or has a disease. So molecular diagnosis is a collection of techniques used to analyze biological markers. So let's look at the biological markers. So anything that can be able to use to say, because you have this, there's a likelihood that you have that. That is a marker. That is what is telling us what is making the difference. So in molecular diagnosis here, now we look at a marker. That marker now can be a sequence of a particular gene. So if you're able to dictate that marker, you can say, because you are here, there's a likelihood that you have HIV. Because you have this marker, there's a likelihood that you have a TB. Because you have this marker, there's a likelihood that you have COVID-19 or every other infection. So it is a replacement of the old techniques, which you generally know. Of course, you saw what happened during the COVID era. Everything changed. All the techniques that we knew were put on hold, and everybody moved into molecular diagnosis. And that was used to not only detect the virus, but to also monitor therapy and be able to use it to even tell people to go home when they test negative. So it has replaced the old way of doing things. It is a new thing. Of course, you fed the wave and you now know the need. And that is why I believe you are doing this course. So it has looked at, phenotypic biochemical products and the byproducts that are being produced are now being looked at in a different way at genomic level, at molecular level. So what this is saying is that it didn't take away the concept of what you know about enzymes. You didn't take a concept of what you know about microorganisms. You didn't take a concept about what you know about ABO grouping. But now we're not just looking at color change or presence of this or that, but we're looking at the gene responsible for that activity. So molecular diagnosis has helped to uncover the underlying mechanism of disease and enabling clients to tailor care at individual level. What is your genetic makeup? What makes you who you are? And that is what is being considered now in treating you. Look at what happened in COVID-19. The Africans had this kind of protection that we can't really imagine. But the fact is that maybe our gene had something coding that was protecting us against COVID. In that case, not people had the exposure, but it didn't come down with obvious symptoms. And then some people had the exposure and they came down with obvious symptoms. So now it looks at individual level and what individuals possess. So let's look at what we use these molecular diagnostics to do. One of them is the dictation of and analysis of nucleic acid molecules, which I've mentioned is either the DNA or the RNA, to provide clinical information. That means as a human being as you are, we can look at your clinical sample, look at your DNA, look at your RNA, and it can tell us clinical information about you. So I think before I move forward here, I would like to know your field so that I can know the examples to use and then ask questions specifically as it relates to your own field. So permit me to call names and then you tell me your field. I have Caleb, how are you? Hello?
Hello.
What's your area of specialty?
I'm Doris. I specialize in histopathology.
In what?
Histopath.
Okay, that's fine. Thank you. What of on midday? I hope I got you correct.
Regarding to biochemistry, I aim to specialize in biology.
In what? Clinical chemistry.
Biochemistry, and I aim to specialize in oncology.
Okay, that's fine. So that's in methodology. Okay. Please mute yourself and then we'll continue so that I will know where my examples will fall in. I need to start slide somewhere. Please let me link up one of my lecturers to Now, with there. Hello, can you hear me?
Yes, I can.
Yeah, sorry, I had to log out to enable another Zoom for other lecturers. Sorry about that. Okay, so continue from where we stopped. Yeah.
I did not carry take away plate. If not, it's have put your. I used to take away to party.
Okay, so I was discussing the dictation of what do we do with molecular diagnostics. We want to dictate nucleic acid and provide clinical information. So one of them is to be able to dictate infectious disease by dictating the pathogen responsible for that disease. So this involves searching for exogenous. Exogenous means if it is in microbiology, things that are coming from outside our body, non-human nucleic acids that can be in the human body, especially if it is a microorganism. Then it is used to search for and possibly, quantify specific pathogen derived from nucleic acid sequences in a patient sample. So when you have a urine sample and you have somebody that have a UTI, so you can actually say this UTI is being caused by E. coli because you're able to dictate the DNA of E. coli in that sample. So the E. coli is an exogenous organism now because it might be coming from outside, or if it is in water, if you're looking at a stool sample with somebody who has diarrhea. So that, especially where organisms are not supposed to be part of the human body, if you see them there, they are exogenous, they are coming from outside. So you can use the DNA to dictate such infectious pathogens. So you can also use molecular to be able to dictate specific pathogens that are also infected or has been exposed to an agent, when somebody has been exposed to an agent. So you can also use that to be able to know the pathogen that is responsible for that infection. So invariably what we're saying is molecular diagnosis is specific and it can lead you to a particular level when we're dictating. You can say you've been able to dictate not only a species in case of microorganism, you can dictate a strain in case of animals or a parasitic infection. You can dictate a clone, you can dictate a variant. Like you have the COVID-2 is a variant of COVID-19 that is infectious. HIV-1, HIV-2, Mycobacterium tuberculosis, and the multi-drug resistant TB, Streptococcus species, and the MR multi-drug resistant Staphylococcus species, E. coli species, and they have the hemorrhagic E. coli, enteropathogenic E. coli, and things like that. We can also talk about serotype, especially the salmonella infection. We can also have genotyping in case of different genes or single isolates. These are different ways you can dictate a pathogen. It can dictate the species, the strength, the clone, the variant, the serotype, genotype, or a single isolate as a whole. So some kids can actually have a sequence that can dictate both the species, strain, clone, variant at the same time. Some can only be to dictate the species causing the infection. So apart from detecting pathogen, molecular diagnosis can also be used in what you call genetic testing. So I'm sure all of you must have heard, at least if you don't know much, you would have heard about testing for forensic testing, is genetic testing, testing for diabetes, things that have to do with the human diseases, leukemia. So you search for endogenous, that means your normal gene has been able to somehow alter itself, either through exposure or congenital abnormalities or difficulties or wrong separation of cells during the time of fission of the fetus. So such abnormalities can now lead to a disease, what we call a mutation, a change in the normal genetic sequences. In that case, what you're looking at is what we call genetic testing, and you are searching for endogenous cells, that is whole derived nucleic acid, using molecular diagnosis techniques. So this encompasses all fields of laboratory medicine. Then here we are targeting a single variant gene. So we can look for a specific one gene, especially in people that has Down syndrome. So their mutation is as chromosome 21. So you can target detection of abnormalities at that chromosome 21. And if you're able to see it, you can say, oh, this person has a Down syndrome because the person has a gene responsible for this. Same thing as in breast cancer, you can actually dictate a single variant gene for BRCA1 gene responsible for breast cancer. And they can say this person is having this gene, the gene has been amplified in this sample, showing that the person has a variation in the breast cancer cells that is showing that the person has breast cancer. So this single gene test look for any genetic change in one gene. And then when you have genetic changes in more than one gene, they were talking about gene panels. So that is why you see cases like a panel can now dictate for 2, 3 genes at the same time. For those of you who did COVID-19 testing, you can see, can remember that it has multiple single genes together to give you what we call like a multiplex, looking at multiple genes at the same time. One of them is to know that the person is having COVID. And then the second one can be, is it COVID-19, SARS-CoV-2? And then another one is it actually a virus that is in there? So those are the different variants, and then the clone, where we're talking about the beta and different clones of COVID-19. So you can also do what you call hematopathology studies or dictation. In that case, you can look at abnormalities in the cells, the normal body cells. Example, those who have blood
I can't hear you again. No. I can't hear you again.
I hope I've not lost my audience. So, oh, they are here.
Welcome back.
Again at today.
Welcome, ma.
Okay. Oh. All right. So I'm still just at the beginning. We've not even gone far. So we're looking at. I hope it doesn't interrupt again. We're looking at. We've mentioned pathogen detection. I've mentioned genetic testing. We're looking at hematopathology testing in terms of blood and things that has to do with blood. Can you hear me?
Yes, ma'am.
Yes, ma'am.
Yes, ma.
Are you seeing my slides?
Yes, ma'am.
Yes,
I
ma'am.
can see.
That's fine. So I think we are done with the hematology. So I'm moving down to
Okay.
the nucleic acid-based test. So when you hear about nucleic acid test, you now start to what does it mean? So nucleic acid test is any test that has to look at the deoxyribonucleic acid and the ribonucleic acid of microorganism that may be associated with a disease or a cell that may be associated with a disease. There are three broad types of assay that can look at this assay. So you are looking at hybridization technique, amplification techniques. Then you have some assay that's used in epidemiological investigation, example, such like strain typing and identification. But in molecular technique, I told you the best, one of the words you can actually grip on at the beginning is the RNA.
No. No, then you should have to put food for.
I think someone needs to move.
If I continue from what I'm seeing, I was saying that in molecular diagnosis, we talked about the tools. The tools have to do with the RNA or the DNA or the protein. We're also going to talk about the technique. Is it hybridization or amplification, or you're looking at some genotyping activities? So confirm that you can still see the slides so that I continue. Can you see my slide, please?
Yes, Caleb can see.
Yes.
Let's look at the hybridization techniques. Hybridization has to do with coming together of complementary base pairs. I'm going to draw a complementary base pair for you in the next slide, and then you can see how they complement each other. This technique is actually used to identification. In what we call hybridization techniques, you can actually use that using the denatured single-strand DNA to be able to carry out the test. Point. Okay, so when you are looking at of techniques, you have a labeled single DNA probe, what you call a probe, which is unique to an organism being tested, and then it is added and hybridized with, and a hybridization take place when you have two sequences that are complementary to each other. So if they are able to hybridize means coming together, they can now bound together. But if they did not come together, the probe will now be bound, will not be bounded, and you can now watch that one away. So the ability to now these two sequences binding together is what you call a hybridization process. And in some techniques, the moment the sequences have hybridized complementary to what is complementary to each other, you can now use things like fluorescence or dials or change in color to be able to see that amplification or hybridization has taken place. So let me also go to the next stage, and then we'll look at it together on what do we say happens when complementary genes have hybridized. Remember that this technique is used for identification, not for dictation, because it is not as sensitive as amplification method. So let's look at hybridization method in its context. In situ hybridization is hybridization that is taking place inside the cell. So a single-strand DNA or RNA sequence is called a probe. Is allowed to form complementary base pair with a DNA or RNA present in a tissue or a chromosomal samples. So we all know what DNA is. Of course, we know a DNA that is double-stranded. So this is a DNA. Can you see that?
Yes.
Okay. So if that is a DNA, and then when you heat it up, it separates, right? The double strand separates into single strands. So this is 1, and this is 2. This process by which the DNA has separated into 2 is what you call denaturing. So when you denature the DNA, the double-stranded DNA will now be denatured into a single-strand DNA. So this is the process of denaturing. So this is for the DNA. Okay. Then we said there's a probe that is complementary to the sequence of interest. So what happens? This probe now can be something you can acquire commercially. You know, you buy it like a kit, like a reagent kit. So the probe now will also have a come here. You can see that this is a probe. This is the DNA. The DNA is double-stranded, the probe is double-stranded. When you denature the DNA, it forms into 2 strands. The same thing when you denature this probe, it also forms into 2 different strands. So if DNA, this DNA strand here is similar to the sequences found in the commercial probe, it can bind to it. Then if it is not similar, it will not bind to it. So when we have a complementary probe, which you can actually determine ahead of time, when you separate the DNA and then the probe separate, they can now bind together, complementary. The ones that are the same will just bind together. So you see what happened. This is our DNA. This is our probe. And the 2 of them has bind together, just like when you hold your 2 fingers to clip them together. This is what happened there. So because this probe has found a complementary DNA and they banded together, this banding together is what you call hybridization of a complementary DNA strand. Now, when this happens, this is what we call label. So this can be a fluorescent, a chemical, or a radioactive label attached to the probe, so that the moment that binding takes place, that thing will fluorescent. Just like when you do agglutination reaction, antigen-antibody reaction in serology, the moment the antigen binds with the complementary antibody, what happens? Agglutination is produced. And that is the same thing as what happens here. So they bind together, and then the chemical or the radioactive compounds can now reflect light to say, oh, I've seen my complementary strand, and then they can bind together and show light. Any questions so far?
No.
Okay, so this DNA now can come from your sample, and then the probe can be your reagent kit that is on the bench you're working with. All right, let's look at amplification. Amplification is a method used to improve the sensitivity of the nucleic acid testing technique. So you can have what you call target amplification, probe amplification, signal amplification, or combination of the above. So in your real-time PCR, you actually have make use of a probe and then a primer. In that case, you can actually have both a target amplification and a probe amplification taking place together in a real-time PCR. So the target amplification will target the gene of interest, and then the hybridization will now complement together and show a fluorescence that can say, oh, I've seen the complementary base sequence. And that is what happened in that machine. So hybridization is a part of polymerase chain reaction and a part of Southern blotting. So let's look at this. This is what I use in explaining PCR in its simplest form. And I pray that you flow along with me. So yeah, PCR takes place is a molecular technique that is being used in the lab to be able to now amplify or the gene of interest. When you amplify that, you have multiple numbers of a gene of interest, then it can do downstream activities too. So that's the essence of PCR. Polymerase chain reaction to amplify one single gene to 2, 2 to 4, 4 to 8, 8 to 16, 16 to 32, and things like that. So it takes place in about 3 stages. Stage 1 is what you call denaturing. And I've been able to use the last figure that I have to be able to show you how a DNA with double strands can be heated and it becomes denatured. So denaturing in PCR takes place at about 94 to 96 degrees centigrade. In that case, you have a DNA in your clinical sample, which you've extracted, you heat it up, it's separate. Then after that, we have what we call annealing, which takes place at a temperature of about 68 degrees centigrade. In that case, if it is a PCR, we're using a primer, the primer will now anneal with the complementary sequence to start the initial reaction. And then after that, the third stage is what you call elongation. You now elongate the base sequences. So I'm going to demonstrate that, but just know that there are 3 stages of PCR: denaturing, annealing, and elongation. So can someone tell me the 3 different stages of PCR?
Denaturation, annealing, and elongation.
Okay, thank you very much. So what temperature does denaturation takes place?
96 degrees centigrade. 94 to 96.
94 to, so you can pick 94, you can pick 95, you can pick 96. That's fine. Thank you so much. And what of elongation?
At 72 degrees centigrade.
72 degrees centigrade. So let's look at what denaturing means, what annealing means, and what elongation means. So who knows what is the component of a DNA of a PCR reaction? What are the reagents you put together to start a PCR reaction? Who has an idea?
No idea. No idea, ma.
Anyway, no idea.
Yes.
There is no idea. Okay, who again?
No idea, ma.
She says no idea. Okay, fine. What of don't tell me that the next person has disappeared. I hope it's not because I'm going to ask a question. So now we're going to have an idea. Let's look at it. So PCR reaction. Well, how can you tell me no idea? I've been talking about something today over and over again. And what is that?
Nucleic acid.
So how can you tell me no idea?
You said reagents used for
So nucleic acid is not a reagent, I mean.
it's not a reagent. So
What is a reagent?
I don't have a definition for reagent, but I know what reagent is used to.
What is a reagent? Let's look at it. What is a reagent?
Okay,
is water a reagent? Is water a reagent?
It can be a reagent. Yes, water can be a reagent too.
So is your DNA a reagent?
Yes, it is.
So you see, everything that can combine together to form a chemical reaction is a reagent. And nucleic acid, thank God we mentioned it, the nucleic acid is a molecule. Is it not molecule we mentioned from the beginning?
It is.
It's simple. So you're gonna think nano. So don't just answer and say no idea when the idea is there. Now I've dropped it down to know that nucleic acid is a reagent. It has a molecule. And I started from with that. That's the first sentence, the first slide I showed you. Then we're looking at reaction at a molecular level. Anyway, if you've not looked at it that way, that's what it is. So even if you don't know anything, you know that one of the reaction mixture is what? Your DNA, right?
Yes, ma'am.
Agreed?
Yes, agreed.
Okay. So the next one, I've mentioned probe today, right? For hybridization, right? So for PPI, we're using a primer. For amplification, we're using a primer. So the next thing here is your primer. So the primer can be a forward primer or a reverse primer. I will explain what that means with this diagram. So the next thing we have is the nucleotides. You know what nucleotides are?
Yes.
Those are the sequences that makes up the DNA, agreed? So mention them.
Thymine, cytosine, adenine.
Adenine, Abby. Okay. So
And uracil for RNA.
Uracil, okay. Okay. So adenine binds with what?
Thymine.
What? Guanine binds with what?
Cytosine.
Cytosine binds with what?
Uracil in RNA.
You know. Adenine binds with what? It's T that binds with you, so I don't know what to say. So T also binds with, sorry, this is a T here, not A. And this is an A here. Is that okay? So what I've just done, I've formed is a genetic sequence. Do you know that?
Yes, ma'am.
What I've just drawn now is a genetic sequence. So all those grammar we're speaking, it's just what I've just done. So this strand, AB, has binded with what? The complementary strand. So let's look at it. This particular strand, right, has bounded with this strand, is it not?
Yes.
Are they complementary to each other?
Yes.
Yes, ma'am.
Okay, so that's just exactly what we're saying. So now in chemical reaction, your DNA, this is just like a DNA now that have normal, complete normal sequences. This is what it's supposed to be naturally. So when I now want to do a PCR, what I'm going to do is to denature this DNA. Denature it. So when I denature it, I will now get a commercial kit or a commercial primer that has the same complementary sequence to bind with it, not my original DNA. So let's see what happened here. So now this is what you call the nucleotides. This AGCAT is the nucleotides, and they are just, you can get them single, you know, to join together. So I can actually join an A with a T, G with a C. Do you understand?
Yes.
Those are the molecules in molecular diagnosis. So these are the molecules in molecular diagnosis. So now, these are what you call the nucleotides. So in a PCR reaction, you have 1, the DNA, 2, the primer, 3, the nucleotide, and then you have the Taq polymerase that is going to join them together. So let's look at what happens here. When you separate, you heat up this, and it separates. It is called what? When you heat up this at 95 degrees Celsius and it separates, it is called what?
Denaturing.
Beautiful. So it becomes denatured. So when it becomes denatured, so this is 1 and this is 2. The 2 of them separate into 2. So I have my commercial reagent, which is the primer. This is the primer now, which was here. We'll now look for a complementary sequence on the natural DNA that we have separated, maybe from your sample, and then bind with the complementary sequence. So for this strand, the primer is also in form of forward and reverse, just like your DNA is in 2. So the other side of the primer now will also come to this 3'. This is 3', this is 3', this way, this one go this way, this one go this way. And then the second part of the primer will now bind to the second strand of the DNA. You see what happens, the red and the green. So when it binds, we still have some blocks, sequences that have not been fixed. We now use the nucleotide now that we've already added into this reaction, will now come and fix. If this is an A here, this will be a T. This is a C and a G. They will all bind and fill up this and become separated. So you now see this is the primer which started the reaction. This is the nucleotide which completed the reaction and continued to extend. So this is the denaturing, this stage is the annealing, and this is the elongation. And then after the elongation, this our single strand DNA here now has become 2, you know, a double strand. So it can now start another reaction. And this one now also form another 2, this one another form another 2. So our 1 here has formed 2 here, our 2 here has formed 4. The moment we hit this one, is going to give us 8. Any reaction? Any question?
Yes, I have some questions, ma.
Okay.
Okay, my first question is that how do we do the specific use PCR analysis? How do we do the specific sequence of?
I can't hear you. You said?
Hear me now.
Please, your line is breaking. I can't hear you.
Can you hear me now?
Please repeat the question. Anybody that understands him should please explain to me. Anybody understand it?
I've typed the question. I've typed it. It's in the chat.
Okay, let me look at it. How do you know the specific sequence of primer to use? Okay, so primers are predetermined. They are predetermined, if you can hear me. Predetermined means for you to be able to do this primer, you must have known the sequence of interest. Then when you know the sequence of that you want, like if I work with microorganism, if I want to get a primer now, number one, I can design a primer based on the sequences of my organism. If I've sequenced the organism, I can design a primer from that. Number two, I can go to existing database and look at primers that have been used before to amplify my gene of interest. So when I get the sequence of the primer, I can now send it to the company that prepares primer. Then I can buy the primer. So it is predetermined. If you want to look for, let's say, Mycobacterium tuberculosis, there's an existing primer for TB. You can now either buy a kit that contains the primer already, or you can send the sequence to a commercial company that are responsible for doing primers to do the primer for you and send to you. Then you can dilute, aliquot, and use. So in summary, primers are predetermined. You can design a primer or use the existing primer that has been designed already. I don't know whether that is clear.
That's,
Huh?
that is very.
Okay, so when you come to the lab, I will show you a primer, the one that has been, it comes in the lyophilized form. So it also comes with the book that we use in diluting it. All right, thanks for the question. Any other one?
Yes, I also touched last one.
Okay, let me see. For essential purposes, are they used primarily reusable? No, they are not reusable because you already added them into the reaction, and then if they find a complementary gene, they will amplify it. But if they don't find it, they will not be involved in the reaction. However, if you now, for that period when you're amplifying, the primer is also going through a process to be able to see if you can see a complementary sequence to bind with. And if you did not, you see anyone to bind with, that's the end of the reaction. So you measure the quantity to use, which is about 0.2 microliters for one sample, the small size. So what you do is you take the quantity you need for that particular reaction. So if you're working with 10 samples, you take primers enough to amplify the gene of 10 samples. So there's a way to calculate it to be able to know that they're using enough primer for the reaction that you want, not excess primers. And then when you have an unused primers, they form what you call primer dimers, which you can see on the gel if you're using the gel electrophoresis to see your product of PCR. You will see all that when we do module 2, looking at challenges of results and what challenges you can get and how to rectify them. Okay, can I go ahead?
Yes.
Okay. So now let's look at the PCR. I've just tried to explain to you PCR, but we'll still look at it during the practical. You're going to do it by yourself and you understand the concept. But that's just a summary of the way I explained to it. So what is involved in molecular diagnosis? We've mentioned either from tissue cells, samples can come from any of those ones, or culture cells. And then we make sure you can extract a DNA or RNA if you're doing a nucleic acid analysis, and then you can do a PCR. So what happens after PCR? So PCR reaction can lead to cloning and sequencing. It's especially directly cloning direct sequencing. It can also lead to amplification method, a sample allele-specific PCR for human genotyping. You can also use your PCR to be able to do electrophoresis method to be able to analyze the amplified amplicons, or you can actually use your PCR directly to the pathogen, especially if you have a probe or a well or a comb that has actually said that they have the primers in there. You can now use that plate to do a microarray analysis, and when the PCR has finished, you can be able to see if there's a change to say that you have been able to dictate your gene of interest. So there are so many things you can do with PCR. So PCR is like a central hub of molecular diagnosis. For you to be able to work with any gene, you have to amplify to see. from one that gene. When you have a larger number of it, then you can do what you call downstream post-PCR activities, which includes cloning, sequencing, amplification method, electrophoresis, and you can actually do what you call what do they call it? Two-step PCR techniques or nested PCR. You finish one PCR, you add another primer, it takes you to another PCR for you to be able to get a product that you can say, I've been able to dictate my gene of interest. So different types of PCR methods that are available in the lab. You have real-time PCR, you have conventional PCR, you have multiplex PCR, you have single-plex PCR, different names of PCR, RFLP, and different kinds of PCR analysis. We're going to look at that in the next slide. So difference between probe and a primer. Remember when I was talking about hybridization, I mentioned a probe, and when I was talking about amplification, I mentioned a primer. So probe is used to detect the presence of a specific DNA fragment in a mixture through the hybridization with a double-stranded DNA. And then primer is used in the initiation, remember the word, initiation of the polymerase chain reaction by hybridization with a single strand DNA. Of course, you saw how I demonstrated the probe, double strand, this way, the probe was used both ways. And I also show you how we did an initial denaturation and then the primer that started to anneal to a single strand DNA. And that hybridization takes place when two complementary strands comes together and then they become amplified. So this is the difference between the primer and a probe. Some reactions can use both probe and a primer. They hybridize and then they amplify. So target amplification requires that the DNA to be tested for that we have what we call a number of copies of the DNA is increasing. You saw that we started with one DNA strand. We got 2, from 2 we got 4, and then from there it keep on increasing. So target amplification is like a PCR reaction, and it's used to amplify something found in such a small amount that without PCR you cannot be able to dictate it. So example here has to do with things that have, in the case of pandemic, they were able to dictate the flu season with the implementation of the E-plex respiratory pathogen panel. In that case, multiple primers are used to know whether somebody has a flu or what you call advanced form of respiratory infection like COVID-19. So that is the SARS-CoV-2 is an example of target amplification. And then things like HIV, TB, cholera, that method is also used to detect them. So these are examples of the molecular techniques, which I told you that I have about 12 that I usually teach on, but there are so many other ones online when you start reading about them. So the one we've talked about now is the polymerase chain reaction, which is a conventional reaction. Someone is going to give you a lecture on PCR, but I try to give it from the beginning because if you understand that any other lecture that you're going to get in terms of the real content of PCR, difference between real-time and conventional PCR, you can be able to grab that. Then these are other techniques other than PCR that can be used in molecular diagnosis. So we have DNA sequencing, like the Sanger sequencing, single gene, next generation sequencing for multi-genomic analysis. They call it metagenomic analysis. Then you have whole exome sequencing, in situ hybridization techniques, Egyptian microarray, mass spectrophotometry analysis, molecular cloning, gel electrophoresis, micromolecule blotting and probing, specific nucleotide analysis, spectral karyotyping imaging. These are all different kinds of molecular methods that you can employ. So we look in all these things, we talk about diagnostic kits. So these are the kits that contain this reagent that I've just mentioned to you. All of us counted the reagents together. So just know that any diagnostic kit that is supposed to be used for molecular should be able to have the positive and negative control, it will be able to have a primer or a probe, should be able to have Taq polymerase that is supposed to use in joining those sequences that we say are being separated when it's at denaturing. Then it should be able to have a nucleotide too, which is a building block to be able to balance the sequences that the nucleotides that makes up a particular sequence. So some of those abnormalities that can be tested, including things like Down syndrome, is the chromosomal conditions where you're able to detect the gene involved in chromosome 21. We also have Klippel syndrome, happens in about 1 in 1,000 males. Additional X chromosome, that's where the gene is involved. We can also look at sickle cell, the big globulin, that is at chromosome 11, is where that gene alteration takes place. We can actually use molecular to detect that. They will look at things like ovarian cancer, Lynch syndrome. These are the genes, the sequences, or the primers that can be used to detect. Like if you're talking about breast cancer, ovarian cancer, we're looking at BRCA1 and BRCA2 gene. So if it is Lynch syndrome, you're looking at MLH1. Then things like Alzheimer's disease, to have PS1, PS2, and APP, it can be used to detect that disease. So you have to get a reagent that has primers specific for this reagent, for these sequences. If it's cystic fibrosis, we have CFTR. And then if it's in dimuscular dystrophy, we have the trophy gene that can be detected. So you find that each disease or each disorder, which is a non-infection disorder, must be a genetic abnormality in terms of mutation at a particular point in the chromosome of the gene. And you can actually have a primer or a probe to detect that gene. And then the abnormality can be tested and be identified in the lab. So these are some of the equipments you use for molecular diagnosis. It's a heating block. Heating block is like a block with a hole where you can put your tube. Is that example the 1.5 mil or 2 mil tube that is being used in molecular? The volume here are actually small compared to the volume when you're using 15 mil tube for centrifuging. Reactions in molecular diagnosis usually in microliters, simple sizes, and some of the activities are measured in nanograms. So this is the PCR. This is my the kind of PCR I have. It's a great PCR, very user-friendly, fast. I have a horizontal electrophoresis apparatus for doing electrophoresis. This is the centrifuge, horizontal centrifuge. Analytical balance to measure the reagents because they're actually small volume. We use about 1 gram of agar, 0.5 gram of agar to be able to separate the PCR amplicon. Then we have microcentrifuge that uses the 2 mil instead of the normal 15 mils you have on the bench. Then you have a photo documentation system that takes a picture of your amplified DNA that has been stained, and you can see the bands. Then you have a lot of reagents, the oligonucleotide primer, which we have mentioned, the Taq polymerase, the DNTP, the master mix, precision enzyme, DNA ladder or marker, agarose, micro.
I'll pack your hair for you.